It's time to go ahead and get started. And first, I'd like to start by introducing myself. I'm Rob Burgess, Chief Business Officer for Sino Biological. And I would like to officially welcome our speakers and all the attendees to the next installment in our series on COVID-19 immunodiagnostics. And the title of this particular seminar series is Cytokine Biomarkers of Inflammatory Response in COVID-19 Patients. Just a few housekeeping issues before I introduce the speakers. Everyone, all the attendees will be muted throughout this particular conference and the seminar, but you will be allowed to ask questions at the end via the chat box. So if you have any questions, I highly recommend that you type them into the chat box, and then I will act as the moderator and I will ask those questions to our individuals who are our lecturers today. In addition to that, at the conclusion of this seminar series, shortly you will get an email with a link and you can therefore download the full recording of these seminars. So with that, I'd like to introduce our first speaker from Quanterix. It is my pleasure and honor to introduce Mr. Kevin Rusovsky, who is the President and Chairman and Chief Executive Officer of Quanterix. Kevin has been with Quanterix for a number of years, and he is actually one of the founders of the company. And he currently serves as the CEO and Chairman of the organization. And in 2017, he successfully took Quanterix public in one of the most successful IPOs for a life sciences tools company in recent history. Prior to joining Quanterix, Kevin was the president of Life Sciences and Technologies at PerkinElmer and the CEO of Caliper Life Sciences and the CEO of Zymark Corporation as well. In addition to that, I want to mention some specific awards that Kevin has received. And specifically, these are the Lifetime Achievement Award for Leadership and the E.G. Bailey Entrepreneurship Award, both issued by The Ohio State University. Kevin holds an MBA from The Ohio State University and a BS in Mechanical Engineering from the same university as well. And I want to mention that he also received an honorary doctorate from Framingham State University for his vast contributions to the life sciences and personal medicine industries. The next individual in our speaker categories is Dr. Dan Sycamore. Dan is currently Vice President of Accelerator and Pharma Services for Quanterix Corporation. Dan has over 25 years experience in the biopharmaceutical, vaccine, and cell gene therapy industries. And he's held many high-level executive positions, including with companies like Bristol Myers Squibb, Wyeth, Merck, Sanofi Pasteur, Frontage Labs, and GlaxoSmithKline. In addition to that, Dan has worked with the World Health Organization, the National Institute for Biological Standards and Controls, and the US CDC, FDA, and EMA as an advisor and an expert. Dan holds a BS in medical technology from Northern Michigan University and a doctorate in microbiology and public health from Michigan State University College of Medicine. And he also did his postdoc in infectious diseases at the State University of New York at Buffalo in the School of Medicine there. And then finally, our third individual involved in today's lecture is Dr. Dara Duffy. He will not be joining us today, but I wanted to say a little bit about his background. He currently leads the Translational Immunology Lab at the Institut Pasteur Paris and is a co-coordinator of the Lab Milieu Interieur Consortium. So with that, I will now turn this over to my colleague and our organizer, Dr. Jen Liu, who will bring up the video presentation.
Hello, everyone. This is Marissa Swayze, Vice President of Public Relations and Marketing at Quanterix. We'd like to thank you all for joining. And lastly, we'd like to mention and give thanks to our webinar sponsor and also our collaborator, Sino Biological. Quanterix has been working closely with Sino Bio on SARS-CoV-2 related projects, and we have used Sino SARS-CoV-2 antibodies on our SIMOA platform. We've obtained promising results. Some of Sino's antigens and antibodies have also been used to manufacture FDA-approved COVID test. We thank Sino for their partnership. And with that, I will turn it over to Kevin Rosofsky.
Thank you very much, Marissa. I too would like to thank Sino. Really exciting to get to know them very well, and they are going to become, I think, a really important collaborator with us as we continue to build out our menu over the following few years. There's a lot of important opportunities, we think, to work with Sino. So I'm real excited today to talk about the innate immune system. This is not a Powering Precision Health Summit webinar. I'm really excited to be doing this in cooperation with all of the people that was mentioned by Marissa. We're going to start off by me just providing a high-level overview of our company and the things that we're working on, primarily in the immune system area, both the innate immune and adaptive immune. But also, we're doing some work in COVID in the antigen landscape that's pretty exciting. And we continue to monitor NFL, which has now found its way into COVID with people losing taste and smell, having some neuronal death measured by our NFL, which has been pretty interesting and important. Dan Sikkema will follow me up with a discussion around the dysregulation and our ability to survey and see early on cytokine storm and be able to actually predict it. And then finally, our real keynote speaker today is Dara Duffy from Institut Pasteur, who's going to be talking specifically around the interferon alpha activity and some of the exacerbated inflammatory responses that we're seeing to COVID-19 in COVID-19 patients. And that dysregulation is triggering, in some ways, cytokine storm, and he's going to describe how measuring interferon alpha can play a key role in that overall interrogation and surveillance of the cytokine in the innate immune system. To start off, we've got this exquisitely sensitive technology in Quanterix, which allows us to be able to see 1,000 times greater the sensitivity. And that sensitivity can be deployed in a lot of ways, not just looking for low abundant biomarkers and proteins. It can also be deployed to dilute samples to improve specificity. It can also be used to go after home care sampling, where you may be just looking at a dry blood spot. We also have been able to show a lot of quantitation at very low levels of quantitation. We can see the entire range, and we also can multiplex and not lose a lot of sensitivity because we have so much sensitivity in the headroom. We did make an announcement 3 weeks ago that we've now found a way to go another 100x with our technology, this single molecule technology, which would allow us to be almost 100,000 times greater sensitivity than other technologies, which again can be deployed in these other ways that we mentioned, dilution being a really important one. On the right-hand side, the ability to transition healthcare from seeing disease late and taking a lot of invasiveness through surgeries and renal spinal taps into early and non-invasive through dry blood spots or through saliva or even venous draws, being able to see exquisite levels of these proteins that translate into a lot of correlative ability to disintegrate and discriminate various clinical disease pathologies. This next slide basically is just showing the instruments that we sell. The HDX is the one that we've just recently rolled out about 3 quarters ago. It's a very fast growing. A lot of the serology assays that we're rolling out into the market is going to be done on the HDX. We can run about 1,000 samples a day. The SRX is also selling very rapidly using the same bead-based technology as the HDX. It's just that it's an instrument that only does the detection, doesn't do the sample preparation like the HDX, which is fully automated. And now we got the planar, which is really finding its way in this landscape of cytokines, where we can run 10 plexes. We call it coreplex on the cytokines and even run some chemokines. Very good economics, very good sensitivity, and it's very high throughput. And we think someday it's going to evolve across into home monitoring and helping us run large panels from dry blood spots to allow us to see biomarkers long before disease the pathology of disease starts to transcend the individual. So we think it's a big breakthrough opportunity. We sell the assays as well. And we have our own services like a CRO where we've already run nearly 200 trials, phase 1, 2, 3 drug trials, and do a lot of studies for customers in our accelerator group. It's a great way to get started with us, as well as later on running major studies. One of the key things we've done in the last 4 years is we've allowed the ability to recruit patients in cohorts earlier in the disease cascade, many times even before symptoms, to allow the drug to have greater efficacy and really treating the disease when it's very early stage. And doing it with lower dose has also lowered toxicity. The net effect of that has increased the probability of a phase 3 success after a phase 1 success by 300%. And you can see that we've actually got 51 instruments now in CROs, and we continue to evolve that CRO positioning and feel it's going to be an important part. And again, we have our own CRO too, and continue to run drug trials for customers. We just want to show on this slide that the publications behind the scenes validating our technology, we're now almost to 1,000 third-party peer-reviewed publications, and the number of biomarkers is over 300 now that we can run. 80 of them are off the shelf, and then there's a lot of homebrew that gets run. And economically, our company continues to grow very rapidly. It's very consistent, rapid growth, which is somewhat of the hallmark that we've been able to create. And it's across all components, whether it be our services, our instrument placements, or the consumables, which now are nearly 50% of our revenue. This is an important growth opportunity for the world, because each one of these instruments that we deploy are working on very important advances in various disease pathology. And you can see on this slide, we have customers now from the research institutions through biopharma companies, the major ones, 24 of the top 25, as well as CROs. This is a very significant penetration in just 3 or 4 years. And we didn't even have any revenue 4 or 5 years ago. So this is a very rapid growth because we're transcending and changing the way drugs are getting developed with this technology. During this COVID period, we said we would keep focusing on neurology, where we just launched last week. I think it was nearly 700 people attended one of our PPH seminars on launching p-tau, phosphorylated tau 181, which has specificity for Alzheimer's and can differentiate away from frontal temporal dementia and other types of dementia. It can give you the specificity in blood. And this is a breakthrough that we already are pretty sold out on that particular assay, which we'll start shipping at the end of July. So in addition to that, though, we really wanted to bring a lot of focus to COVID, our accelerator in China. And so on the COVID front, you can see on this slide that many of the technologies can only see the tops of the peaks of the innate immune system, as well as the adaptive immune serology, and even the antigen. The time that they can see it is really dictated by their sensitivity. With our technology, we actually can see most of these curves because of the single molecule sensitivity, which does allow us to quantitate. It allows us also to do home care through finger prick. And in addition, we think it's important to see these antibodies or the antigen earlier, long before potentially even symptoms. And many times in COVID, many patients don't even present with symptoms. And so seeing these different biomarkers early, we think is very important. And we're going to spend a lot of time talking about the left-hand side, the innate immune system today, the cytokine storm, which we're able to see with our exquisite sensitivity. And many of the researchers around the world are deploying it. And we're trying to roll up the information on all of the immune responses, as well as the antigen, which we've just begun to see in blood. We've got a new assay that we're working on. We would hope in the next several months we will be rolling this out. We would like to see it in saliva as well, so we're working on getting some samples in now to see the antigen, like our HIV P24, which was 1,000 times more sensitive. We'd like to be able to see someday the antigen in saliva to make it a much easier test to deploy and to have a lot better sensitivity with the specificity. And then the adaptive immune system, the serology, we are rolling those technologies out by the end of this month, actually. So we're pretty excited about being able to see IgG and quantitative. We have researchers around the world that are supporting us with sample sets in all of the hot zones. And some of these collaborations are shown on this slide. We're very proud to have such amazing researchers collaborating with us. And many of the new technologies that we're deploying, we will test bed them out into these partners. And right now, I know Henrik Zetterberg, who just spoke last week with Kai Blenlau on the p-tau 181, he'll be running some of the serology in his HDX over in Sweden. And we're doing the same with Charlotte over in the Netherlands, doing the same with LabCorp out on the West Coast, to begin seeing from a research basis, RUO basis, exactly all of the capability that the serology assay has. And then we will roll that out broadly, we hope by the end of this month, to anyone that has an HDX. So now moving into cytokine, we're very excited today to have Dare Duffy and both Dan Sikkema describing in more detail what we're able to do here. But I just wanted on this slide to show you their HDX and the SPX, and you can see the panels that we can provide today. And then there's a lot of homebrew as well, but seeing this interferon alpha, the first spike earlier, is key. And we're actually now working on 8 different subtypes of interferon alpha because of our sensitivity that further will stratify what this interferon alpha signal means. And so we would like to think in the next, maybe by fourth quarter, we'll be in a position that we could start to roll this out. It's a collaboration we have with Servier. And in this case today, the interferon alpha could be low early and trigger IL-6 and also TNF alpha spikes, which then later on the interferon alpha catches up to and spikes up. And so there's some interesting signatures that lead to the pro-inflammation that you're going to hear about in today's talks. I wanted to show this too, because even in neurology, there's a lot of key inflammation markers. And this was a study that was just issued last week showing how capable our technology is versus MSD and Luminex. And I think, you can see that the key here is our limit of quantitation is so low that 100% of the data points for all these key markers for, in this case, it's post-traumatic stress disorder. There's some neuronal inflammation through cytokines that we can measure. 100% of the measurements are above our lower limit of quantitation, and that allows the full spectrum from healthy to disease to be seen. And this is a very good paper, if you haven't seen it, to further augment and showcase what we have. And in the area of serology, you can see in this slide that there are ELISAs out there today, but the ability to quantitate and get answers from 1 microliters and from dry blood spots and see them really early, and then see the full spectrum signature of all 3 isotypes across 4 different antigens. These are the types of capabilities that we're trying to bring into serology over the next several months in a research basis. And we will consider an EAU as well with the FDA for patient samples and results. But at this point, we're going to try to roll out the RUO by the end of this month of June. And I did want to also continue to point out that NFL plays a role, even in COVID, with us seeing those that have lost taste and smell having neuronal death, and being able to see the spikes of NFL in blood has been another key assay that's been being deployed. And we also know NFL now seeing its way into almost every neuronal degenerative disease, as well as concussion. And these number of publications now is growing at almost exponentially. And several drugs in the area of MS have now been approved utilizing our NFL as a secondary surrogate endpoint versus the gold standard MRI. And it's a much lower cost, much faster to see the evidence of the disease in blood with the technology. And we are running 2 different trials. One's an analytical validity, where in 17 different sites around the world, we got great CVs across all those sites. And then on the right, clinical validity, looking at what is the NFL level when you're healthy. And then we have a large population study that's going to establish that line, and then that will help us for showing MS and other diseases that go above the line. And so with that, Dan, I'd like to turn it over to you to start a deeper dive into the area of our innate immune system surveillance.
Yes, great. Thank you so much, Kevin. So I'm going to talk today a little bit about traditional vaccine approaches and how SIMOA can kind of get us to the next level. So this first slide I'm showing here really describes how we've done historically with vaccines. We've done a really great job with the development of prophylactic vaccines. We have many vaccines for childhood as well as elderly diseases. And I think the whole COVID situation has really driven a lot of innovation into the vaccine world with all of these different approaches that all of these different companies, nearly 100 different companies working on vaccines, and about 8 or 10 of them already in clinical trials, some phase 2s and phase 3s even starting within the next month. And I want to draw your attention to here that therapeutically, to treat an existing disease is a much different approach. There are so many different viruses that can cause cancer. We now have checkpoint inhibitors. We see a lot of development ongoing with the use of these checkpoint inhibitors with appropriate antigens to try to get rid of things like herpes or hepatitis B or hepatitis C, human papillomavirus, something that someone already has. They're beyond the point of prevention. Next slide, please. The history of vaccines in the United States, you can see they've been tremendously successful on a per dollar spent basis. There's never been a more successful medical modality in the history of the world. And we're sort of embarking on a new frontier now with COVID, where we're not just going after a particular age group or a particular geography. We're essentially needing to develop a vaccine that can be deployed worldwide. So the numbers are going to be even larger. And as I was talking to on the previous slide, looking at oncology vaccines and trying to have therapeutic vaccines that cure something that already exists, again, the scale of that is going to be so much different than anything we've ever encountered before. And if we can get immuno-oncology and oncology and these cancer vaccines to the level of success that we've enjoyed with vaccines, just think of how great the world will be. When you look at vaccine biomarkers, historically, people are looking at toxin neutralization for diphtheria and tetanus, and viral neutralization is really the key for something like COVID-19. And there are many examples of viral neutralization assays that exist today, and these are cell-based assays. And in the case of COVID-19, are run in a BSL-3 laboratory, not really scalable. So our goal at Quanterix is to develop serologic assays based on immunoglobulin G or even subpopulations of the IgG and A and M that are especially effective at neutralizing viral infections and correlating them to those cellular diseases, so that we have a broadly deployable assay that can be deployed globally and worldwide rather than only in highly specialized, very small laboratories with very low throughput. So examples of immunogenicity that have been used for correlates of protection, you'll see on this slide, these are historical data from World Health Organization and other organizations. Typically, you derive a particular antibody level or a particular functional antibody level that correlates with protecting a population from disease. This is still to be determined for COVID-19. The viral neutralization, as I said, is considered the gold standard, but you can see that many of these other diseases above have evolved to a immunoassay, an enzyme immunoassay, an ELISA, where they may have actually started out as a toxin neutralization assay or a viral neutralization assay. So it certainly is possible. It depends on the size of the trials and the duration of the trials, but these are all the types of things that we're trying to accomplish with the Quanterix technology. So why do people measure antibodies? You'll hear that that's been in the news a lot lately. It's been all over the news. How do we determine whether a person is immune and whether that immunogenicity is durable? People measure antibodies for a variety of reasons historically, and a natural infection may be very different than a vaccination. The natural infection is going to have antibodies against a wider variety of proteins within the virus, and not just specifically those that are to the vaccine antigens. So those will be some fundamental differences. So when people are enrolling their clinical trials for these vaccine studies, they're going to want to know if a person has had a prior exposure. Again, having a highly sensitive assay to determine whether a person has been previously exposed and has already prepared or antibodies against the virus. And we're going to want to also know how long those antibodies last, what's the durability of them. There may be studies where in the elderly that they're giving the vaccine, I'll talk about that on the next slide, where they're giving flu vaccines and other vaccines at the same time. And then ultimately correlating those serum antibodies to what we call a correlate of protection. And I'm going to talk for a moment also about antibody maturity, avidity maturity, and memory, and why those are important in the vaccine world. So when you look at childhood vaccines, you can see that there's a variety of different schedules. Different countries of the world use different vaccines from different manufacturers. They give them at a different time schedule, so it gets very complicated very quickly. And then in the case of adolescents, now we have vaccines against meningococcal disease, and we have vaccines against human papillomavirus. And in the elderly, as I mentioned, they may want to only go to the doctor once and get their shot for shingles or their pneumonia shot or their flu shot. So people that are doing these vaccine studies are going to have to take all of these different types of things into account. It may be a standalone vaccine initially. Again, we don't know. It's too early in the process of developing the vaccines. But ultimately, they would like to administer these vaccines as combinations rather than coming back individually for separate shots. And it's not just how much antibody a person makes. What's the isotype? So Kevin talked about IgG, IgA, and IgM against 4 different antigens. We also know that with the COVID infection, initially people make an IgG3 response. That's very unique. That's not very common in a lot of different bacterial and viral pathogens. And over time, so people make antibodies that first week of exposure. The antibodies change in the second week and third week and fourth week post-exposure, so the antibodies mature. And functional antibodies can take even longer to develop. They can take 3 or 4 weeks to develop, or they might require several different shots. So 2 different immunizations a month apart. How well do those antibodies persist? We measure them right after 4 weeks after immunization, but what about 3 months later, 6 months later, 12 months later? All of these things are being accounted for in vaccine clinical trials going forward. Is there memory? If you give them another shot a year later, does it jump up very quickly, or does it act as if it's a primary exposure and you're starting all over from scratch again with your immune system and it's going to take 3 or 4 weeks? And is memory fast enough? We know that the COVID infection, people say the incubation period is roughly 2 weeks. So if you can mount an immune response, and memory responses typically take 3 to 5 to 7 days. That's great. The example that I have here about Haemophilus B meningitis, from colonization in the throat to a very tragic, or perhaps even as severe as death to the child, can occur in as short as 24 to 48 hours. So that's why we give childhood vaccines at 2, 4, and 6 months of age and another shot in the second year of life. We have to keep the threshold of the antibody levels high enough for that first 2 years of life while their immune system is maturing, so that they don't get these really rapid onset meningitis type of organisms. I think we're good. It remains to be determined, but I think we're good with something like COVID to have a memory response. So even though antibody titers will go down over time, if a memory response is robust and sufficient, that should protect people against subsequent challenges from the real virus. And this is just a slide I took from New England Journal of Medicine many years ago, showing that a lot of different viruses and a lot of different natural exposure to viruses gives a very long-term, lifelong immunity. Whereas you see on the lower right-hand side, tetanus and diphtheria, that's a shot that everybody is recommended to receive every 10 years. And that's because the immunogenicity to those recombinant antigens, those purified antigens, is not as durable as necessarily a natural infection with some of these viruses or a live virus vaccine. And as we're moving forward with the Quanterix technology, it's also very important that we work with regulators, and we're doing this routinely, working with FDA, Centers for Disease Control, the World Health Organization, all of these different types of organizations, to engage them in this technology, educate them about the technology, differentiate technology from historical means. And we've been actually invited to the FDA several times, and I was astounded because the comment was, "This is wonderful. Can you educate us on this? We always feel like we're behind in technology, and people are submitting data to us on technologies that we're not familiar with." And they've actually created entire educational programs within the FDA to bring in outside vendors to educate them on new types of technologies and their benefits. So that was actually very rewarding. I'm going to talk for a moment here the use of a mycobacterial stimulant in a pancreatic cancer study. I didn't know what this was initially. I was looking at interferon gamma in patients that are treated with a standard of care for pancreatic cancer, and they used what they called a non-specific immunostimulant, and they called it IMM 101. When I looked it up, I found out that it's essentially proteins from a bacteria very similar to tuberculosis. And you'll see on my very last bullet point here that the BCG vaccine may actually influence healthy innate immune responses. And we've worked with a collaborator at Harvard, and she's published on this extensively over the past decade, using the BCG vaccine to actually reverse or modulate diabetes type 1. And we've actually found out, because of all of these COVID studies, that countries that still routinely use the BCG, like South Korea and Japan, had perhaps 50% less deaths than they had in other countries that do not use it. So there may actually be benefit to using BCG, even though it's not directly a COVID vaccine, but it may have safety effects or protective effects, or actually help the immune system, the innate immune system, which Dara is going to talk about in a few moments specifically. A couple slides very quickly here. HIV, these are using latency reversal agents where the virus is hiding so successfully in people they can't detect it anymore by PCR. A new class of drugs called latency reversal agents chases the virus back out. And they felt it was very important to use our technology to measure virion expression by protein synthesis and not PCR to actually measure de novo virus coming back. And a little tip to Sino Biologics here, we used their antibody pair from a typical ELISA, had a sensitivity for detecting the COVID nucleocapsid protein at about 48 picograms per ml. Simply using those excellent antibodies on our technology actually improved the sensitivity to 15 femtograms per ml, essentially a 3,000-fold improvement in sensitivity. And going forward here also, we did the same thing with the dengue viral program. And dengue virus is a very unique virus in that second infections are much more severe than first infections. So having a highly sensitive IgM assay, in this case, 10,000 times more sensitive than diagnostic kits, was actually very beneficial for identifying primary infections with the dengue virus versus secondary infections. And the same thing for Clostridium difficile. The nucleic acid test actually tells you whether the bacteria is present, but the bacteria is not what hurts the patient. It's actually the Clostridium difficile toxin, which is expressed and secreted from the bacteria. So it's separate from the bacteria. So a person could theoretically have the bacteria, but not be expressing the toxin. So again, just like botulism toxin and other types of toxins, these are present at incredibly low rates and require a very, very sensitive assay to measure them. And traditionally, they're measured in stool. Being able to measure them in blood is much more of a simple approach and actually determines if they have a biologically relevant level of the concentration of that toxin. And I just also want to note on my sort of getting to my last slide or two here, that we're not limited to just antibodies and antigen proteins. We can actually use nucleic acid probes as capture molecules within the technology to detect nucleic acid at the single molecule level. And we're measuring directly what is there at the nucleic acid level, not what's amplified, so we don't have the problems with contamination during amplification. So we are seeing some collaborators use our technologies in ways that we hadn't really thought of initially, but using them very successfully and getting PCR level sensitivity at the nucleic acid without having to use any amplification. A wide variety of samples that have been used, so people come to us with all different kinds of issues, and we like to solve their problems. As we said, we have a CLIA certified laboratory. We do a lot of assay development, custom applications for those clients. So they come to us with an existing assay or a need for an assay, and we work with them to develop the most sensitive, best assay for their particular application. Cardiology, you'll see on the right-hand side some of those same cytokines that are involved in the cytokine storm with COVID. So there's been a lot of evolution in cardiac monitoring of people. And again, as Kevin said, being able to find things before they're symptomatic. You don't want to wait until you're shoveling your driveway in February. You want to know if you have ongoing cardiac disease and get treatment. So asymptomatic detection of disease is preferable to symptomatic detection. I think my last slide here talks about parallelism. And again, Biogen tested 9 different assay technologies and looked at them for these 4 cytokines. And you can't really do parallelism if you can only get one dilution to be able to measure your sample and everything else is below the limit of quantitation. As Kevin said, having extra headroom on that sensitivity allows us to do additional dilutional linearity studies and determine the parallelism of our unknown samples relative to our calibrators. And this has been a big issue with the FDA over the past several years in Crystal City meetings, where people want to implement parallelism studies for biomarkers, and the main reason being that biomarkers typically use a recombinant calibrator, but you're measuring native proteins, and dilutability is a key parameter in establishing parallelism, and we've been able to do that very successfully. So this is my last slide, and I'll turn things over, but SIMOA is really here to help enable clinical validation, moving those research programs and development programs into clinical validation, and helping develop new biomarkers with greater sensitivity for disease management going forward. Thank you very much.
So we'd now like to invite Dara.
Hi. So thanks, Kevin, Dan, Marissa, and Elizabeth for the invitation to present some of our COVID research, ongoing work at the Institut Pasteur, where I lead a translational immunology lab. And really our primary research interest is to understand variability in immune responses. Why are any two people have different immune responses and have different responses to infection? So that's really the fundamental basis of many of our projects. And when the COVID-19 pandemic arrived, it seemed really pertinent in terms of the strategies we apply to understand variability in immunity, in particular to infection. If you think about infection with SARS-CoV-2, there's different outcomes. There's a high percentage of asymptomatic people, so people whose immune response can clear the infection without showing any major symptoms. And there was this nice meta-analysis from Eric Topol's group at the Scripps recently, where they looked at a lot of these big cohort studies all around the world and really saw consistent high levels of asymptomatic infection between 30 and 45%. And they looked at some of the reasons for this, and this is still ongoing work, so it's hypotheses rather than shown, but really some of the prominent ideas for protection and asymptomatic infection are either the presence of cross-reactive coronavirus antibodies, or cellular immunity against some of the other coronaviruses that are out there. It could be due to differences in expression of the cellular receptor for the virus, so mainly ACE, but there could be other players implicated in that are coming out in all of the ongoing studies that have been published every few days now. The genetics is starting to give some interesting results. There's this intriguing association with non-type A blood that's come out in a few studies, so it still needs to be confirmed that it's not due to any underlying compounding factors. And one GWAS study that has reported some variability in in chromosome 3 that appears to be potentially associated with the receptor for the virus. But that's in terms of asymptomatic infection. What's different in individuals that don't succumb to severe infection. But of course, more people do display symptoms, but still the majority of people that do display symptoms manage to control it and have a mild or moderate infection, where the estimate numbers still a range between 45 and 65% of people infected with the virus. They could have symptoms that are equivalent to a severe flu, with days, weeks, even months of unpleasant symptoms, but they don't require critical medical help. Unfortunately, for those people that do, and here the estimate is between 5 and 10% of infection cases that go on to be classified as severe or critical. This seems to occur in a two-wave passive. There's initial presentation of symptoms that are mild to moderate, and then in a second wave, there's a severe worsening of clinical symptoms, really in the respiratory tract between 9 and 12 days later. And this leads through into the high death rate of 0.5 to 1%. And some of the strong risk factors for severity, this is pretty consistent and well understood now, as really old age, male sex, and high BMI. But we still don't really know how those factors are impacting an individual's ability to moderate the virus or to go on to develop symptoms. So as immunologists, we really wanted to look at, well, what's different in the immune responses of these individuals that may lead to these different patterns. And some of the objectives we had with this approach was, can we identify biomarkers to stratify patients based on their immune response? Can we identify the key immune pathways that are required for protection to the virus to give us clues for new therapies? And we just heard a lot about vaccines. Can we identify correlates of protection to guide the vaccine design and eventually vaccine deployment, because we still don't really have good correlates of protection against SARS-CoV-2. So to ask some of these questions, we established a study at Hôpital Cochin, so in collaboration with many different people. Some of the key people here are listed here, from Hôpital Cochin, from Pasteur and AP-HP in Paris, and Imagine Institute. And this was back in March, the last 2 weeks of March, really at the beginning of the peak of the pandemic in France, where we managed to recruit 50 COVID patients with a mean age of 55. There was a sex bias towards men, and they were harmonized over days 8 to 12 post symptoms, because that seems to be really the key moment for the development of severity or critical disease. So all of the patients were in that 8 to 12 days post symptoms period. So this is the nasal swab viral load within these individuals, just to make the point that the differences in their outcome wasn't due to differences in viral load. So really, it's the ideal setup for studying what is different in the immune responses between these different categories of patients. So in its first study, we looked at the immune responses in the blood, different technologies. So CyTOF, which is a mass cytometry-based approach where you can analyze circulating immune cells in the blood. We did gene expression by nanostring. So this is a hybridization array approach where we quantified up to 600 key immune genes. And then for cytokines, we use both SIMOA and Luminex. So we've been using SIMOA for a number of years. And in this study, we used our own homebrew triplex, where we measure interferon alpha-2, interferon gamma, and IL-17. We also used a Quanterix triplex of IL-6, TNF-alpha, and IL-10, and then a homebrew singleplex for interferon beta, one of the other major type 1 interferon responses. So in the past, we've been measuring all of the subtypes of interferon alpha with a pan-interferon alpha antibody. Unfortunately, we don't have the right to use those antibodies anymore, but we've been using this interferon alpha-2 specific assay, which correlates pretty well in the vast majority of patients with the pan-alpha responses. In parallel, we ran a Luminex, so this is a 45-plex high-performance assay from R&D. And I'm going to go through each of these different technologies, what we've learned from them, and the color code for the patients is really all the same, and it goes from left to right in terms of healthy controls, moderate, severe, and critical. So the first, what I'm going to show you is the CyTOF data. So it's been reported in severe COVID disease, there is lymphopenia in the blood. This is confirmed here by the clinical assays. So the idea with the CyTOF was to go for a more unbiased approach and see if we could drill down into the subpopulations of immune cells and see what is different. So here with CyTOF, we measured over 30 parameters, and this is a t-SNE plot, so a dimension reductionality approach, where you can capture all of that data in 2 dimensions as plotted on this t-SNE map, and then color code for the different immune cell subtypes. So if you look at the t-SNE map for healthy control, the moderate and the severe and the critical, you can start to see some differences, namely down here. So this large presence of the gray, this is differences in monocytes. And up here, you can see differences, which are T cells. But we can obviously drill down and look at those individual plots and numbers of the cells. And here you can see a decline in CD3 cells, decline in CD8 T cells, but also a decline in NK cells with critical infection. In parallel to seeing this decline of circulating lymphocytes, we also saw an increase in exhaustion markers and apoptotic markers on those cells. So here you're looking at an increase in the expression of PD-1 on the CD4 cells, increase in PD-1 on the activated CD8 cells, and TIM3 on the NK cells. So these are all activation exhaustion markers. So meaning that these T cells are been driven towards exhaustion. We also saw higher levels of annexin-5. This is an apoptosis marker, both in CD4 and CD8 T cells, suggesting that these T cells are undergoing apoptosis. In parallel to the loss of T cells, we also saw an increase in circulating B cells and plasmablasts. So here's the increase in CD19 B cells and an increase in plasmablasts. And this occurs in parallel to the production of antibodies. And this is confirmed with an increase in B cell activation and plasmablast differentiation genes that were measured by gene expression. So switching from the cellular assays to transcriptomics, I mentioned we ran a nanostring, their human immunology panel. This is a clustering heat map showing you the differences you can see with increasing disease severity, really an increase in many of these immune genes as you move towards critical disease. So we wanted to drill down into this to see what genes could tell us might be driving these differences between these patients. And this is a PCA analysis. You can see in black, these are all the healthy controls, and really a difference along principal component 1 between these healthy controls and the severe and critical, with the mild to moderate split across that. So if we look at what's driving that PC1, we did GCA enrichment analysis on the gene sets, and we could see it's really differences in immune responses as expected, increase in innate immune signaling, TLR signaling, MHC1, really all these pathways are going up with infection, as you might expect. But more interestingly, if we now look at PC2, you can start to see slight segregation of the red, the critical, and the severe and the moderate. So this was really more interesting. Now, if we drill down into what's driving PC2, here there's less, but we still came out with 2 pathways, a type 1 interferon signaling and a type 2 interferon signaling. So we wanted to look at this in more detail. And now look, this is a heat map of the ISG genes. So the genes induced by type 1 interferons. And here it started to get a little bit more complicated in that we saw an increase in the critical cases of genes involved in signaling, such as the receptor, IFNAR1, JAK1 and TYK2. So these are the molecules just downstream of the receptor. However, for some of the key antiviral ISGs, such as MX1, IFIT1, IFIT2, these were downregulated in critical disease. So really starts to get a bit more nuanced and complicated in what's going on in these interferon signaling pathways. So we wanted to obviously measure the protein and see, well, are all of the patients secreting the same levels of interferon alpha protein, the key antiviral cytokine, and this is where the SIMOA assays came in. So we measured interferon alpha protein by SIMOA. Here you can see the healthy controls. We have a strong induction in the moderate cases, but really a striking decline as you progress towards critical disease. We also complemented this by measuring interferon activity. So this is a cytopathic assay that measures the functional ability of plasma from these patients to inhibit viral infection in an ex vivo assay. And this paralleled with the alpha-2 protein expression and also the ISG score. So this is a cumulative score of 6 key ISG genes in the blood. And interestingly, these ISGs correlated nicely with the interferon alpha protein as measured by SIMOA. So really appears to be a consistent defect in type 1 interferon responses in these critical patients. So this was a single time point. So the question now is, well, so before I go into that, we also went back to the cellular data and looked at the plasmacytoid dendritic cells. These are the major circulating immune cells that secrete type 1 interferon. And indeed, they were downregulated in critical disease, and this correlated with the interferon alpha-2 expression. So really, many layers of the type 1 interferon pathway seem to be dysregulated in critical COVID-19 infection. So I mentioned about the timing. So all of these patients were between 8 to 12, initially 8 to 12 days post-symptomatic. We did manage to get some samples that were at a later point. And if we split the different groups across those days, where they're grouped between 8 to 10, 11 to 13, and 14 to 17, we can see in the moderate cases, although the numbers are low, the interferon alpha protein remains high. But with severe and critical cases, it either goes low or is low throughout. And then in a smaller subset of patients, we managed to get a retrospective sample that was prior to them being admitted to ICU. So this is now graphed into stable. This is prior to the development of critical disease and when those patients were admitted into the ICU. And you can see that low type 1 interferon preceded clinical deterioration and actually had a pretty strong odds ratio for association with development of critical risk. So that's in terms of the interferon. And then just the last part, we know about the cytokine storm in COVID. So we also looked at many of the chemokines and cytokines that are associated with that. We could see again a lot of this large red in this heat map in the critical cases is due to an increase in those key inflammation markers that's mapped here in terms of the NF-kappa B signaling pathway. And then we confirm that at the protein level, sorry, at the cellular level first, an increase in circulating neutrophils, no changes in circulating classical monocytes, but a severe downregulation in non-classical monocytes in parallel to this increase in inflammation. And then confirming that at the protein level, many groups have shown IL-6 using more clinical-based assays. We confirm that with SIMOA, where we really get this strong distinction between healthy groups where IL-6 is really low, and the moderate and the severe and the critical. And then more interesting, TNF-alpha, that is sometimes more upstream of IL-6 and is proposed to be targeted with anti-TNF therapies. Here there was a strong significant difference, even though the levels were much more lower and subtle. And here, this is really the power of SIMOA, that you can only really pull out these differences because of the accuracy you have at this low picogram or even sub-picogram per ml. Interestingly, I1 beta was no difference. This was measured with the Luminex assay. We want to go further with this in SIMOA because the receptor antagonist was significantly higher and critical. And we still don't fully understand what's going on there because there was no induction of I1 beta protein, at least measured by the Luminex assay. So in conclusions, what I hope I've convinced you is that there really is impaired type 1 interferon responses that are associated with increased disease severity in critical cases of COVID-19. This is in parallel to increases in inflammatory pathways and NF-kappa B signaling, and we also identified perturbations in homeostasis and cell death pathways. And this has given indications for new treatment strategies to really target and boost the interferon response while potentially in parallel inhibiting inflammation. And these particular results have inspired an ongoing clinical study, an interventional one with treatments to really address that. We do need to be careful because the timing in these responses is critical. We need longitudinal studies to address that. And I mentioned some of the limitations here. It's a single time point for most of the data sets. It's a homogeneous patient population, and it's only in blood. So we are addressing that with some ongoing studies, working with other collaborators, in particular going into the nasal swabs to the infected site. So working with the team of James De Santo, also integrating the antibody responses, working with Olivier Schwartz and Hugo Moque. Clearly, antibodies are important, in particular to understand what's good for a vaccine response, and extending this beyond a homogeneous adult population to look in pediatric patients and elderly patients where there's both less and more severe cases of infection. And then going further in these perturbed immune responses, looking at the stimulated immune response, working with teams at Trinity College Dublin to really see in more depth how these type 1 interferon and inflammatory responses are perturbed by stimulating the response of these patients. So I'm going to stop there with the key last slide of many, many people involved in this. I listed all the co-authors on this preprint. It's currently in peer review, so we hope it will be published soon. And also for some of the ongoing work with the different teams at Pasteur, and I've highlighted in bold the individuals at Pasteur that are working with me and my team to enable this study, and of course the financing from the French ANR, the Institut Pasteur, and the LabX Militaire Consortium, which I'm co-coordinating at Pasteur. So I'll stop there and happy to take any questions and join in the discussion.
First of all, I want to thank Kevin, you and Dan, and certainly Dara for just a wonderful seminar and presentation. Since we only have a few minutes right now, I want to jump right into the question and answer session. And anyone who has a question, please type it into the chat box and we'll do our best to get to it. And so I think I'll direct the first question to you, Kevin, if you don't mind. And I apologize if you've already answered this. There was a lot of information there. But one of the technical questions that we got, and be sure you don't reveal any trade secrets here. One of the technical questions that we got was, how does the Quanterix technology differ from that of Singulex, for example? Can you comment a little bit on that?
Sure. Singulex basically is a little different type of approach in the way that they measure, but the two most important differences between their technology and our technology, number one is that most of what they did was in a centralized lab where you would send your technology or your samples into them. They've subsequently built some technology that was acquired, I believe, by Millipore that it now provides actual instrumentation. But typically you're looking at singleplex, looking at one analyte. And beyond that, the second most important difference is the sensitivity. I think their sensitivity approached ours and maybe it got to within 10x. So probably of all the different analytical techniques we saw at their Singulex had probably gotten the most sensitive beyond us. And we've now subsequently gone another 100x in our sensitivity. So that was a bar that we had, prior to our announcement 2 months ago. And I think most of the company is now been broken up as well. So it's no longer, a company where you can get their technologies, but the sensitivity is pretty important, not just for seeing low abundant analytes, but it also allows you to eliminate matrix effects and get answers from much smaller samples, like from dry blood spots, or from less invasive samples like saliva or urine. We had a podcast yesterday of a prostate cancer panel in urine. So the sensitivity can really reveal itself in ways other than just looking for low abundant markers. And the key thing is seeing disease earlier before symptoms.
Right. And since I've got you here, Kevin, I'll just ask a very related question. I thought it was a pretty good one, actually. Is cytokine storm was mentioned several times, and the question is really, why is there such an emphasis, at least for COVID-19, on sensitivity? Because a lot of cytokines are already expressed at relatively high levels, and is there a chance that you're seeing false positive with your system? Would you mind commenting on really the need for sensitivity for looking at cytokines for COVID-19 positive patients?
Sure, and Dan might want to comment as well on this one, but in general, there's different subtypes of cytokines that are now becoming more and more important. And one of the more important cytokines that we're measuring is interferon alpha, which ends up being one of the early spikes, early elevations, and function to that particular cytokine, interferon alpha, at lower levels or at higher levels can trigger the storm. And so I think it's a key measurement from baseline, and it's the ability to compare to the baseline that requires the sensitivity. And again, I think what we're starting to see is people want to get these answers from dry blood spots or finger pricks. And then once you start to go after less invasive samples, their concentration of those markers is at much lower levels. So it's a combination of being able to see it from baseline or being able to see it from a much less invasive sample. I don't know, Dan, if you wanted to add to this.
I think that's exactly right. And interferon alpha is typically in the very low picogram or even femtogram range. So it's not like some of the other cytokines that are very abundant, these very early signals, and the virus even seems to have mechanisms to suppress many different pathogens suppress innate immune responses, and TNF alpha is sort of their during their getting their foothold and starting the infection. That's one way they protect themselves, is by kind of escaping or dampening the innate immune system.
Great. Thank you, Dan and Kevin, for that. And I had another good question here. There's a question about a defined cytokine panel for COVID-19, and maybe there are even unique panels for the different severities of the disease. Is there a specific cytokine panel? And if there is, and let's say, for example, you had a cancer patient who also may have been infected with COVID-19, how do you distinguish, or how do you statistically tell the difference between the two?
So Dan, I'll comment first and maybe you want to follow up, but I do think that one of the areas of sensitivity, once again, that can be deployed is multiplex. If you have a lot of different analytes you're looking at simultaneously, you normally give up some level of sensitivity. But if you have a lot of extra headroom and sensitivity, it allows you to do a lot of multiplexing. And so we have a 10-plex that we run on our SPX that has most of the key cytokines in it. And each person's a little bit different, but IL-6 and the interferon alpha end up being a couple, IL-3 of the more important ones. And so we have different panels that cover those cytokines, but I wouldn't say there's anything that we've launched that's specific. And most of the panels that we had built were for oncology. And then, our primary focus on oncology was looking at the immune system and looking at immunotherapies and their effectiveness and trying to see movement and upregulation of the key cytokines from the medicine itself and its comparison to baseline and timing. So once again, I think oncology has a lot of similarities because you're looking at that immune system. Dan, did you want to comment on the statistics that was being asked in the oncology question?
Yeah, I think you covered it very well. And we are seeing at least several different mechanisms with COVID where some people respond to an anti-IL-6 treatment and some do not. So there are some distinct differences in certain types of patients that benefit from an anti-IL-6 treatment. Some benefit from an anti-TNF alpha treatment. People are trying other anti-cytokine treatments, anti-IL-17A in particular, and others as well that are currently marketed drugs. So they're repurposing these drugs and trying to better understand and looking at a multitude of these cytokines at different time points. We can see which ones are going up and down and which ones are heading in the wrong direction. And people are coming up with algorithms based on, so it's very early research, but as we're getting these clinical study samples from these key infected areas and we're getting longitudinal studies, it allows us to figure this out. So that's kind of just touching on what Kevin, just adding to what he said.
Great, and that leads into one of the next questions, which it sounds like you may have some data on, but patients that are on anti-inflammatory medication, do you have data on that? Are there any interesting findings where you see the cytokine or chemokine panels and profiles change?
Yeah, so Dan, you might want to comment, but after this, I would say that it's been intriguing to me that there are different, there are some vaccines, BCG, that have shown different levels of immunity or immune, translation that there is some evidence, and Dr. Dennis Falkman is doing some work up on this to, see what that might actually revealed, where they think it's creating some level of immunity towards this COVID-19. I don't know if we've seen any data that shows any of the current immune suppression drugs and the role that they've played in like a prophylactic or preventing the disease. There was a lot of speculation being raised by our president at one point, but I'm not sure if I've seen any solid studies that have warranted that as a preventative measure. And we also have seen ACE inhibitors playing a pretty interesting role, which could be blocking some of the infection moving into cells. And people that were on ACE inhibitors, there are some trials going on right now trying to see, why it is that the population, those that take an ACE inhibitor, seem to be much lower, almost like it could be a preventive measure. But more science is needed. Dan, do you have any evidence?
Yeah, Kevin and I have talked about this a fair amount. When people are taking TNF inhibitors and some of these drugs, the amount of free TNF becomes 100 or 1,000 or 1,000,000 times lower because it's all being bound by the antibody drugs that bind TNF or bind IL-6. So we had the first assay in the world to be able to measure a free TNF in the presence of TNF inhibitors. So having this extra sensitivity, not only down to baseline, but well below baseline in some of these drugs allows us to measure how much free drug is present, even in the presence of some of these antibody inhibitors. So that was just one extra thing I wanted to add on top of that.
Great. Thank you, Dan and Kevin. And I think we're just about out of time. I just wanted to ask one more specific question. It's really related more to the business model and success of Quanterix. And Kevin, maybe you can wrap it up by answering this. Is how many instruments do you have in the field? What's the breakdown in research versus clinical use? Maybe what's the list price of an instrument? And one technical question is, how long does it take typically to perform an assay on the instrument?
Yeah, we don't provide a lot of granularity. We're a public company, publicly traded, and so we have to be sensitive. We don't provide selective information, but overall, we're close to about 500 instruments around the world that have been deployed. Most of the instruments are in the category of HD1 and HDX, which HDX was launched really just about 2 quarters ago, and it's a fast-growing technology, a little bit more sensitive, higher reliability, provides some additional opportunities for multiplexing, et cetera. That instrument, by year end, we did project that half of the HDs that we have out there will likely be HDXs. The SRX, that HDX runs about $200,000, and the HD1 is similar in pricing, but we don't only sell the HDX now, no longer the HD1. The SRX came on the scene about 2.5 years ago. It probably is rivaling the number of total installs that we have for the HD series. And that price point is more like around $75,000 per instrument. And then the final one is SPX, which is doing a lot of the multiplexing of the cytokines. That also is around $75,000. And I would say that most of what we serve is research use only, but with SARS, there's a whole lot of new assays that are creating a lot of attention. We have a new antigen capability, measuring the antigen. We had a researcher this morning revealing some incredible data to us. It was a homebrew based on some technology that we're developing for a kit that's actually allowing that kit to create a lot of antigens visibility even in dry blood spots, as well as saliva. So we're working hard on that. As the serology, we have an opportunity to move that into the clinic. And I think if you look at the number of LDT and CLIA labs, there's probably about 50 instruments around the world that really could get into a clinical opportunity, and then probably another 150 instruments that could be moved into an LDT site on a current site. So, you know, not much today, but it could ramp up as we try to help support serology and antigen testing for the world.
That's fantastic. Well, congratulations to you and Dan and others on your success at Quanterix. It's really a fascinating and wonderful platform. And Quanterix is a great partner for Sino Biological, so we really do appreciate that. And we appreciate your lecture today as well. So with that, I believe we're over time now. I want to thank the speakers for their time today. We really appreciate it. I want to thank all the hundreds of attendees that joined us today. We appreciate your time and attention as well. And finally, I specifically want to thank my colleague, Jen Liu at Sino, who coordinated and organized and executed this entire webinar. So remember, this is the second in about 8 webinars. Please check the Sino website for, I think we've got 5 or 6 left, and register for those as well. And with that, I think we'll close the meeting. Thank you again, speakers, and everyone enjoy the rest of your day.